Hello, I'm Sophie Mentzel, a PhD candidate at the Norwegian Institute of Water Research in Oslo, and the study is part of the ECORIS 2050 project, and together with my colleagues and supervisors Merete Grung, Knut Erik Tollefsen, Marianne Steenroth, Roger Holton and Yannick Moore, we are working on a Bayesian network approach for probabilistic risk assessment of pesticides under agricultural and climate scenarios now and in future. We developed the Bayesian network to assess environmental risk of pesticides under future scenarios that incorporates various types of information, climate projections, pesticide exposure models, and toxicity tests. And one of the main benefits of Bayesian network models are that uncertainty of all components can be quantified and that the probabilistic risk calculations can be carried out. So why does this matter? Well, land use and weather patterns will shift along with the way water and food resources are obtained and managed all over the world by 2050. In Northern Europe, this results in an increase in precipitation and temperature, and this may cause an increase in plant disease and insect pests. Also, predicted population increases will change the product demands. These expected changes will alter the agricultural practices, as well as the crop types and the use patterns of pesticides. As current probabilistic risk assessment typically takes into account the variability of species sensitivity with the species sensitivity distribution, it lacks consideration of the variability and magnitude of the exposure to hazardous chemicals and other factors that influence the exposure to or effects of these chemicals. So there is a need to integrate future change scenarios such as climate, application and crop in risk assessment, as well as carrying out a more site-specific exposure assessment. This study explores an alternative approach to probabilistic risk assessment using a Bayesian network modeling. And summing it up, the approach is based on Verdonk et al. 2003 and derives a fully probabilistic risk quotient from exposure and effect distributions. We use the Bayesian network, which is a probabilistic graphical model, and here you can see an example of the Bayesian network. The model contains two parent nodes, exposure and effect, and a child node, risk quotient. And these are connected through arcs, which are basically underlying conditional probability tables, and each node has states with probabilities, and the model is updated with the Bayes theorem. A great example for ecological risk assessment was published this year by Carragher and Barron, and it shows a risk assessment for mercury for the Florida panther. Another example by Lara Grassland-Tatro of a Bayesian network integrating environmental changes such as river temperature and stream cover, together with toxicity and the effects on the abundance of smallmouth bass, is displayed in this slide. You can find the link to this interesting Bayesian network as well. Environmental factors such as soil and site parameters, together with chemical properties and climate scenarios, are linked to the exposure of pesticides in the selected study area by using pesticide fate and transport models, in this case a WISPE model. WISPE stands for the World Integrated System of Pesticide Exposure and can predict and evaluate the potential impacts of pesticides on the environment as it is configured with scenarios containing data on crop, soil, and weather conditions for major agricultural areas. For more information about the WISPE model, you can check out Bolli et al. 2013. In this study, we focus on environmental risk of pesticides in a case study region representative of Northern Europe. And this study area simulated was Syverud, which is in the southeast of Norway. The soil in this representative field site is representative for Norwegian soil, especially in the southeast, and has been classified as loam or silt loam. The concept was developed for several realistic scenarios that were used as an input for the WISPE model. The aim was to simulate chemicals to specify unique pesticide application conditions for different scenarios. For the start, we used winter cereals, as the recent trends has shifted from spring to winter cereals, and we ran the model with current climate data from 1990 to 2015. And for a representative field study site that I mentioned before. Application scenarios were determined by using the assumed worst case application rate and creating 2 additional inputs for 25% above and below the worst case. The model was run with these scenarios for now, and the output of the model are 26 predicted concentrations corresponding to the 26 years over which the model runs. The concentrations were predicted for instantaneous, 24 hours, 96 hours, 21 days, 60 days, and 90 days. These concentrations were used as an input for the Bayesian network, which derives the risk quotient by dividing the exposure by effect distribution and applying safety factors. The general approach for the risk model is explained in more detail in another platform presentation, which is displayed here. The exposure input in the Bayesian network into the Bayesian network is displayed in this figure, and the data was log transformed. The y-axis displays the concentration and the x-axis time since application for each of the 26 years the model was run, and a linear equation was used as an input into the exposure node of the Bayesian network. The preliminary effect distribution used so far is based on Ecotoxicity data, EC50 values. If multiple EC50 values for the same species existed, the mean was used. Overall, only 3 taxonomic groups were collected, 13 species, and the toxicity tests used were only including adverse effect endpoints. The mean of the distribution was 10.6 with a standard deviation of 1.57. The conceptual model for the Bayesian network was developed, and for now we only have application scenarios. The instantaneous concentration together with the time since application determines the time-specific concentration, and for the effect concentration an extra node endpoint is added to differentiate between acute and chronic exposure, and exposure and effect together determine that exposure effect ratio node, and later on an uncertainty factor can be applied that derives the risk quotient. The finalized Bayesian network model can be run by selecting a set of scenarios as evidence, and given this evidence, probability distribution will be updated throughout the Bayesian network. This is an example for the compiled Bayesian network. The worst case application scenarios with 1 day time since application was used to derive the time-specific concentration. And furthermore, an uncertainty factor of 100 was applied, and the Bayesian network then predicts the probability of risk quotient intervals. And in the following, the risk quotient probabilities for the intervals are displayed in a stock bar to enable easier comparison of the results. So this bar chart displays the output for the risk quotient for all 3 application scenarios, for the time since application of 1 day and an uncertainty factor of 10. The differences between the risk quotients are very small, though the risk is higher for a higher application. The second chart displays the output for the risk quotient for the highest application scenario and the time since application of 1 and an uncertainty factor of 1, 10 and 100. Uncertainty factors are applied to demonstrate how the alternative affect the risk quotients, and the selection of an appropriate uncertainty factor should be done by a risk assessor. This approach has the potential to assess the environmental risk of pesticides under future scenarios. It integrates different types of information from climate pesticide exposure models and toxicity testing. Also, it gives a transparent way to display the risk quotient. Further plans for the Bayesian network is to integrate other crop scenarios. We are also planning to run the model with 2 predicted climate scenarios for Norway and other representative field sites that are more representative for growing vegetables and potatoes. This research was funded by European Horizon 2020, and for more information you can visit the following website, NIVA website. And I would also like to point out some of the interesting talks from my EcoRisk 2050 family members that are participating in SciCon2. One of them is Taylor Lane, focusing on the impact of climate change on chemical and environmental fate processes. And the other is Samuel Welch, who is carrying out risk assessment for pharmaceutics. And then there's another interesting talk from a NIVA colleague, Simon Schmidt, who is dealing with conceptual adverse outcome pathways. Thank you very much for your attention.